Okay. Hi, my name is Mira Han, and I'm here to tell you about our experience training machine learning algorithms on enhancer perturbation studies. So understanding how a particular enhancer finds a specific target gene to enhance its transcription is still an open question, and we thought maybe we can learn such rules from the data through machine learning. And the motivation for it came from these first three studies that came out around 2019, 2020, that did CRISPR interference experiments in parallel in K562 cell lines across hundreds of enhancers, and then read out the gene expression of multiple genes. And we thought this is accumulated enough data to train a supervised model for understanding this relationship. The training data that we used was from Gasparini et al. This is the largest CRISPR interference experiment done using multiplexing of guide RNAs. We targeted more than 5,000 candidate enhancers to disrupt, and then they used single-cell RNA-seq to read out the expression of more than 10,000 genes. And if they see lower expression of the target gene in the cells where the guide RNAs are present, then they called that enhancer and the promoter of the downregulated gene as a functional enhancer-promoter pair. So our machine learning is trying to predict those positive pairs where the enhancer functionally regulates the target gene. Unfortunately, there were only less than 1% of the total data that was positive. It was about 600 EP pairs out of 66,000 potential EP candidates, so that was an extremely imbalanced data set. Before we get to our model, this is the accepted model in the field that has two components to understand EP relationship. One part is the strength of the enhancer that can be measured by the H3K27 acetylation. And then the other is the high C contact intensity between the window that contains the enhancer and the window that contains the promoter of the target gene. Unlike the ABC model, which is an unsupervised model, we used supervised gradient boosting to train the model on the Gasparini's data. The features that we included in the model were the variables that were included in the activity by contact model. And in addition, we had several more different kind of histone modification data. And then we had the transcription factor binding assays done by ChIP-seq from ENCODE that was on the enhancer side and the promoter side. We set aside chromosome 5, 10, 15, and 20 from Gasparini as the test set, and we used two additional studies that we introduced earlier as independent test sets. So here is our result. We saw that generally the supervised model performed better than the ABC or the distance-based model. And when you compare the model, let me just take out the laser pointer. Okay, so the XGP are the supervised model. The full model includes all transcription factor features. They generally had about 6% higher precision recall compared to the reduced model without the transcription factor peaks. So that was good, but then when we took this model and applied it to the two additional data sets, we couldn't see that significant increase in performance compared to ABC. So we were wondering what's the difference between these data sets compared to our training data. And these two experiments were targeted experiments. They didn't read out the gain expression genome-wide. Folco targeted about 30 genes, and Schreiber targeted about 200 genes. And justifiably, they targeted genes that they thought were most likely to be regulated by distal enhancers. So we went back to our Gasparini training data and filtered the data in the same way. This time we only included genes that had at least one positive enhancer that were called. And hello? Are we still connected?
Please continue.
Oh, okay. Sorry. Yes. And once we filtered the data set to include genes with positive enhancers, we saw the same pattern where we couldn't see the significant increase in performance compared to ABC. So we interpret it as if you have a gene that has a large effect enhancer that you know is targeting the gene already, then finding that enhancer in the vicinity, you only need Hi-C and the enhancer strength measured by histone modification, and additional transcription factor information did not help in the prediction. If we look at the overall feature importance, it also corroborated the APC model because the most important feature was the Hi-C contact between the enhancer and the promoter. So the way that you these kind of graphs is so red value means high feature value, blue means low feature value. The red being on the right side of the graph means that high feature value predicts positive EP pairs. So this means that high contact is going to predict positive enhancer promoter relationship. And you see that other component of the APC model, H3K27 acetylation on the enhancer, is also ranked very highly in feature importance. But our model also identified some novel insights. One of them was it learned how to predict negative EP pairs. So if you look at these two features, the more lower the value, the more positive the prediction. So it means that if you have these clusters of transcription factor binding peaks on the promoter, then you were likely to predict negative enhancer promoter pairs. And these clusters of transcription factor binding sites were including proteins such as HCSC1 and another protein called MZ. And these are actually activating transcription factors, and they were also correlated with the histone modification mark, which is an enhancer mark on the promoter. So it looks like if your promoter has a very strong enhancer mark and a couple of activating transcription factor binding peaks, then it is a very strong promoter that can activate expression by itself, and it is less likely to be regulated by distal enhancers. And by identifying these negative EP pairs, our model was able to perform better. As you can see in these comparative matrices, the positive prediction in ABC and our supervised model is not very different, but the main performance increase comes from being able to reduce the false positives by identifying negative EP pairs. A couple features that predicted positive pairs or some transcription factor clusters on the promoters and the enhancers. The transcription factors on the promoter side were general RNA polymerases and splicing factors. Transcription factors on the enhancer side were the well-known transcription factor for cell cycle and hematopoiesis, which is what you expect for 0562's leukemia cell lines. And then there was a feature that predicted negative P pairs on the enhancer side. So there were a couple of protein transcription factors that when you see this presence on the enhancer was likely to be negative. But if you look at the actual proteins, these are all well-known proteins that are members of the activated protein 1 AP1 complex. And AP1 complex is actually enriched in enhancer hubs and is a well-known activator protein. So the interpretation of this is that if you have AP1 complex binding the enhancer, that enhancer is more likely to be part of an enhancer hub where multiple enhancers are regulating a single gene. And CRISPR interference on one of those enhancers are not likely to downregulate the gene very much because there are other enhancers that are redundant and robustly activating the gene. The last feature we want to highlight are the density of the enhancers and promoters within the collective landscape. You can see this in raw data of the Gasparini's experiments. If you sort the genes by how many enhancers and promoters are nearby, what you find is that for Gasparini to call it a positive EP pair, if you were in a region where there were lots of enhancers and promoters, you need a higher, stronger free contact. And our model was picking that up as well. So we saw that if you're in a region where you have very many enhancers and promoters, you were less likely to be called a positive enhancer. But interestingly, when we focused on enhancers that have indirect contact with the promoter, these are enhancers that have very low IC contact. There were a subset of enhancers that only showed positive prediction in very dense regions where there were lots of other enhancers and transcription start sites. So we went back to the raw data and looked at the characteristics of these enhancers. And these were enhancers that didn't have the other positive cases because they had very, very low IC contact with the promoter. They had lower enhancer marks, and they reside in regions where they have a lot of promoters and a lot of enhancers. So to summarize, integrating genomic assays such as transcription factor binding peaks with enhancer perturbation studies increase the accuracy of the model for enhancer promoter specificity. And most of our increase in performance was achieved by learning where the CRISPR interference experiments didn't work, by identifying the negative cases where the machine learning was picking up the features of very robust transcriptional activation, either by strong promoters or by robust enhancer hubs. We identified atypical enhancers in high-density regions that do not follow the characteristics of positive enhancers. And there seem to be more to learn about these relationships, but it'll be in the enhancers that have modest effects or enhancers with redundancy or synergistic effects in their regulation. This work was done by my students, especially Dylan Barth is the lead author of the paper, and you can find the paper published in Bioinformatics. Thank you for letting me talk and join remotely.
Thank you very much for the presentation. Are there any questions or comments? Okay, please. Yes, please use this mic.
Great work. So a quick question for you. So now the enhanced promoter interaction is known to be mediated by CTCF gene. So have you ever tried to include CTCF binding site, whether it can improve your prediction accuracy or not? Thank you.
Thank you for that question. Yes, we did have CTCF ChIP-seq in our feature. They were part of the clusters that were predictive, but they weren't as predictive as the other clusters. So it does show up in the feature importance, but in a lower ranking. I think it's because CTCFs generally tend to be at the boundary of these loops, while what we were looking at was ChIP-seq peaks right at the enhancer and right at the promoter. So that's probably why they weren't predictive as much. But it would be interesting to look at the intermediate region between enhancer and promoter and see if there's any signal there, if there is ChIP-seq peak of CTCF insulator within that region, but we didn't do that work yet.